A certain type of sugar could increase the growth of cancers. This is what the highly respected journal Nature recently published. In this study, researchers from Washington University in St. Louis, Missouri, discovered a new mechanism linking a certain type of sugar to the worsening of certain tumors. In sodas, ready meals, and any edible material processed by the food industry, we almost always find the presence of fructose in greater or lesser quantities. The main source that is used is corn syrup because of its high fructose content. Corn syrup is obtained industrially by breaking down cornstarch. It is very popular for two main reasons. First of all, for its very high sweetening power. For comparison, the sweetening power of fructose is greater than that of glucose. The second reason is, unsurprisingly, its very low cost, which contributes to its wide use. Just look at the importance of corn production in the United States. The omnipresence of obesity in Uncle Sam's country is not unrelated to the excessive consumption of fructose and its harmful effects that do not stop at simply gaining weight and metabolic diseases. Indeed, if our plates are not spared by the often discreet presence of fructose, the study published in the journal Nature suggests that fructose could also accelerate the growth of cancers. A small clarification, all the same, glucose and fructose, although both simple sugars, do not follow the same paths in our body. Unlike glucose, which circulates in the blood to provide energy directly to cells, fructose takes a different metabolic pathway. A large part is metabolized by the liver, where it is converted into lipids. In conducting their study, the researchers noticed something very interesting. In vitro, cancer cells exposed to fructose do not show marked growth. This suggests that fructose does not directly feed tumors. However, in vivo the results are quite different. Indeed, in mice, a diet rich in fructose leads the liver to produce large quantities of specific lipids called lysophosphatidylcholines. Not easy. For ease of pronunciation, you will allow me to use the acronym LPC. These molecules, once released into the blood, will significantly stimulate the proliferation of tumors. By providing the bricks necessary for the construction of cell membranes, LPCs will play a key role, and in doing so, they will facilitate the multiplication of cancer cells. Since the 1970s, the massive introduction of high fructose corn syrup has coincided with a dramatic increase in obesity rates in the United States. Today, more than 40% of American adults are obese, and this trend is observed in other Western countries. The study also highlights an indirect but alarming link. Excess fructose promotes the production of lipids in the liver, which not only increases the risk of metabolic diseases, such as type 2 diabetes and cardiovascular diseases, but also the proliferation of cancer cells. Obesity, already known as a risk factor for several cancers, including breast, colon, and prostate cancer, could be partly explained by this effect of fructose on the production of pro-tumor lipids. If we take a biological point of view, what happens? First, and once ingested, fructose will be metabolized in the liver. Fructose is quickly transformed into metabolic intermediaries that fuel the synthesis of triglycerides and phospholipids, such as LPCs. Then, LPCs are released into the bloodstream, and that's where the distribution begins. Lipids circulating in the blood can reach tumors, where they act as growth stimulators. We already knew about the relationship between fructose and LPCs, which has already been studied in research on metabolism, inflammation, and metabolic diseases such as fatty liver, type 2 diabetes, and cardiovascular disease. What's new in this study is the additional pathological link between LPCs and tumor proliferation. LPCs therefore promote the proliferation of cancer cells by providing elements necessary for the construction of new cell membranes, which are essential for cell multiplication. The discovery of the Missouri researchers therefore highlights an indirect but significant role of fructose in tumor progression and raises important questions about the impact of our modern diet. By highlighting the fructose, LPC, cancer link, we can question the expected benefits of fruit consumption. Fruit consumption, as a natural source of fructose and other nutrients, could indirectly influence LPC levels in the body. However, fruits do not directly produce LPC, but they can affect their metabolism via their bioactive components. Fructose from fruits is indeed metabolized in the liver, but it is present in much lower amounts than in added sugars or sugary drinks. As a result, the synthesis of fats in the liver from sugar induced by fruit fructose is minimal and less likely to disrupt the lipid profile, including LPCs. In fact, fruits consumed whole and fresh contain fibers, antioxidants that will have a positive impact on the degradation of LPCs. Here again, it's all a question of quantity and quality. So avoid bottled fruit juices and prefer whole fruits. However, in people with metabolic disorders, even moderate amounts of fructose, including that of fruits, could have different effects. It depends on the overall context, such as the amount of fructose consumed and the general quality of the diet. If we look back at the study, I think that the results obtained should sound like an alarm signal for consumers. I would add that being the first actors in our well-being, it is quite easy to invest on our scale in primary prevention. Here are some tips to remember. Start by moderating your fructose consumption. Avoid processed products rich in corn syrup as much as possible and favor natural foods. It is also important to raise self-awareness about the risks of fructose. This sugar is not only a contributor to obesity, it also plays a potential role in cancer, as demonstrated by this study. Finally, promote natural alternatives. A diet rich in vegetables, with a moderate consumption of natural sugars, leading de facto to risk reduction. This study opens up exciting new avenues for research. The first could be to identify other lipids involved in the development of cancers. Indeed, LPCs may be only the first in a long list of lipids produced by the liver that promote tumor growth. We can also expect potential innovative therapeutic levers that would consist of developing targeted therapies to block the production or action of LPCs in order to slow the progression of cancers. On the other hand, this study was only based on the animal model, and no clinical study has yet been observed on humans. Although the results on mice are very significant, further research is needed to confirm these effects in humans. In conclusion, this new study demonstrates that fructose, often perceived as a simple sugar, reveals a very harmful impact on our health, particularly in the context of cancer. These findings remind us of the importance of making informed food choices and of deepening our knowledge of the complex links between diet, metabolism, and diseases. Thank you for watching this video, and feel free to subscribe and join me on my Telegram channel linked in the description. Share this video if it can be appreciated by others, and above all, take care.